Good morning. First of all, I want to thank the organizers for inviting me to give this opening talk. Let me share my screen with you. I need just some few seconds to get organized. The title of my talk is updating the conceptual framework of the regulation of transcription initiation in bacteria. As we all know, it's a fundamental property of life, of every single organism, to have any gene subject to regulation of the amount of expression. And the very first time we could implement a mechanistic model of gene regulation is with the basic definitions of an operon by Francois Jacob and Jacques Monod in the early 60s. So it's just for reminding us, an operon was defined as a unit of coordinated expression constituted by an operator and the group of structural genes regulated by it. You can see the diagram with polycistronic two structural genes subject to common transcription. And of course, since then, there's been a huge amount of increasing knowledge of detailed studies of different promoters, different transcription factors, the organization of transcription units, regulatory interactions. What I'm showing you here is the growth of curation in terms of number of all these objects through the years in the last almost 3 decades that my group has been working on precisely gathering all this information, organizing it into databases, such as RegulonDB, where you can find updated information, knowledge of all these different types of elements in the genome. And our work is also feeding EcoPsych. Now, in both databases, essentially the current schema that is behind our structure of the knowledge is somehow depicted here with different transcription factors, binding sites, right? The conformation of the binding of the transcription factors with different allosteric metabolites, the organization of the transcription units from individual promoters that are subject to initiation by RNA polymerase with specific sigma factors, whenever known, of course. RNA polymerase itself is subject to allosteric regulation, like PPGPP. And then in some cases, we do have also knowledge about the terminators. Ideally, every single piece of knowledge is gathered in the database with the corresponding type of experimental or computational evidence that supports our understanding, our knowledge. As years have passed and we have essentially maintained the same framework, we may wonder how much are we just preserving this classic model when new knowledge is constantly challenging the framework of our encoding of all this knowledge, right? So to give an example of this high contrast between knowledge in the 60s and our current knowledge, just look at the definition of promoter in 1964 as a sequence between the operator and the beginning of the operon that is required for gene expression of the operon. That was essentially based on mutation knowledge, and compared to our amazingly highly detailed understanding with the crystal structure of RNA polymerase, sophisticated studies, biophysical studies of the different subcomponents of the RNA polymerase, the alpha subunit, the beta prime, and so on, the sigma subunits, where are they binding, what role are they playing in the detailed mechanism of transcription initiation, right? Another example is also the appeared in the. I myself devoted my postdoc to do a comprehensive collection of promoters that are subject to regulation. What you see here in the 90s, what you can see here is a collection of promoters aligned in relation to the +1 of transcription initiation with a specific depiction of the binding sites for different regulatory proteins, transcription factors, right? This is just a subset of the 120 or so promoters gathered in. That was the sigma 70 collection. And this is the smaller collection of sigma 54. And with this initial subset of knowledge before genomics, there was already the beginning of the ability for us to make some general statements. So sigma 54 was clearly a case where there's no necessity of a proximal site close enough to where the transcription factor requires to directly bind with, interact with RNA polymerase, thanks to the bending of DNA, of course, and the fact that sigma 54 can make a stable complex, close complex compared to sigma 70. In this year, in July, we published a work on sort of redefining, reevaluating the fundamental concepts, these classic definitions of operons, operators, regulon, and so on. And with gathering a collection of a group of colleagues, experts from different perspectives on gene regulation, some of them like Steve Bosby and Joe Wade, experimentalists, other people that are more on the bioinformatics side with Jack Van Gelden, Adam Arkin, Gary Stormo, and people, experts on ontologies, gene ontologies, Karin Albeck, and then people more in metabolic perspectives like Bernard Paulson and James Gallagher with ChIP-seq recent studies, myself, and Citlali, a PhD student that devoted a good amount of work and effort on not on using RegulonDB. That was quite interesting to learn because having the collection of the data is not what was necessary. Of course, it was extremely, very helpful, but essentially we needed to go and look to the definitions and search for extreme cases. So the methodology we utilized was in order to update and formalize new definitions of these fundamental concepts was to look for features that are necessary and sufficient for the definition of each of the different objects or components. So bear in mind, a feature is not necessary for the definition, for instance, of a promoter, if a member if a given promoter does not have the feature. And conversely, the feature is not going to be sufficient to define the promoter or a transcription factor binding site if an object has that feature and it is not part of that class of let's say of a promoter or binding site, right? So with this sort of essential logical tools in our hands, we started looking one by one. So here I'm going just to show you, of course, in this short amount of time, some few cases that illustrate the type of work that we did. So this is one example. Motifs, as we all know, are essential to understand the regulation of gene expression at the level of transcription initiation, with motifs identified. What I'm showing you here is on the bottom, the -10, the Tata box in sigma 70 promoters in E. coli, and or Privenon box, who was the first to, with only around 7 sequences then, started identifying a motif, a conserved set of sequences. and then of nucleotides, and some few distance upstream, the -35 box. And of course, these type of patterns are the logical results of the biophysics of the interaction between what you see in this figure of the crystal structure of, of a protein CRP binding to its operator site. So the protein DNA interaction has limitations on the ability, on the flexibility, on the variation of the possible nucleotides, with those that are more conserved, of course, the ones that are being involved, that are involved in the specific recognition interaction with regulatory proteins or with RNA polymerase. Now, so one may wonder, are motifs like those ones just mentioned, the -10, the -35, or in the case of sigma 54, the -12 and -24, are they essential? Are they part of the required in our definition, given the detailed knowledge we currently have, right? I'm showing you here the sigma 70 collection of possible motifs, the core, the discriminator, the -10 or Pribnov box, the extended -10, the -35 box, and then UP the UP for upstream element or elements that are known to interact with the alpha CTD subunit, and then the other ones are essentially interacting with the sigma specific factor. So are they necessary to define a promoter? Well, we think they are not necessary. Some promoters don't have the motifs. I mean, for instance, the -35 is frequently absent in promoters subject to activation. As shown here, the C2 activator sitting around the -35 are providing the chemical elements that substitute for a good -35. And similarly, even the -10 might not be easy to identify in some promoters. And of course, the UP elements, they are also frequently absent. And this absence of motifs, in fact, has to do with the very nature of gene regulation. If you think about a perfect promoter would be strongly binding RNA polymerase and might not be easily subject to gene regulation. So anyway, the bottom line, motifs are not necessary, and they are also not sufficient. Some motifs are present in sequences that don't have the full properties of being a promoter. So you may have, it's been shown, the possibility of having binding of the sigma 54 without any transcriptional activity, right? So motifs are not necessary, and they are not sufficient to define a promoter. This is just, as I said, a short story of a more detailed analysis of several other properties that I'm not mentioning here, and you can find in that recent paper in Nature Review Genetics. The new definition we came about is promoter as a DNA segment that is essential for initiation, for specific initiation of transcription at a defined location in a DNA molecule and recognized by a specific sigma factor. And additionally, a promoter is not always autonomous. That is to say, sometimes the activity requires the presence of an activator, right? So almost every single word was highly discussed, and we had to, because there's a lot of details of knowledge in, for instance, we know that there are different sigma factors that can bind in very much the same region and utilize exactly the same transcription start site. So we propose these are different promoters given the definition you see here, recognized by a specific sigma factor. So if two different sigma factors are recognized in the same sequence, these are two different promoters, for instance. A specific initiation at a defined location. So we know very well that the flexibility of RNA polymerase and opening of the bubble and so on might in some cases it's being shown that the +1 can be, there's more than 1 + 1 option. And so it is a defined location. It doesn't mean it has to be a unique position. So I'm just showing you the sophisticated level of discussions we had to go to, had to go through in order to make the best possible of our efforts to something that is relatively easy to follow, to understand, and that captures the essential of the current knowledge. We don't know how long, by the way, these definitions are going to support new surprising knowledge that might wait for us ahead in the near future. In this slide, that is not a fancy one with no figures, I'm just summarizing the story of the notion of operators to transcription factor binding sites and to our proposal of distinguishing between TFBS, transcription factor binding site, and transcription factor regulatory site. So the story started with the definition of an operator for the binding site of a repressor with the LAC modeling in mind. Shortly after that, emerged the very first examples of positive regulation with the Arabinose operon, and also in Lambda, something that the founders of the concept were not very easily going to accept. But anyway, that was the data, as we all know. Operators then became binding sites for repressors or activators. And the language started to become ambiguous, and different minds using the same words with different ideas in their heads. Currently, they are called transcription factor binding sites. But then with genomics and the new technologies of massive identification of binding sites in a genome, with ChIP-seq, for instance, and RNA-seq, we know by now that many binding sites have no clear role in transcription initiation. No, it doesn't mean they don't have. Maybe new experiments will show that. But in our creation work, we have to encode as much as possible the current knowledge and be ready to modify as new knowledge comes by, emerges. So we suggest to distinguish between, as I said, binding sites from transcription factor regulatory sites. Furthermore, many binding sites have been shown to occur inside the genes, very few of them being really regulatory sites. So what you can see here is a graph of the number in purple of binding sites from, all of these are data from high throughput experiments between binding sites and regulatory sites with these methodologies in some few different transcription factors. So going back to history, an operon was defined by included one operator. And we would know since a while ago that one promoter can be subject to multiple binding sites, to the extreme that some people like me and others have defined modules or phrases, groups of binding sites and transcription factors bound there that are participating in a single mechanism of regulating a promoter. This was the basis of the work I did in the 90s of a combinatorial grammar, the possibility with some very few rules, rewriting rules, and a rich dictionary to be able to generate the whole collection of the then diversity of non-regulated promoters. Years later, people like Terihua have implemented logical models with sort of on and off gates, and very much following the similar type of ideas, but of course with different methodologies and more quantitative approaches. Rob Phillips has, with colleagues, continued the idea of a statistical mechanical model of gene regulation with the aim of eventually having a much more quantitative model detailed understanding of every single regulated promoter. And what you can see here in the figure is sort of the ideas of the precisely quantitative description of the different combinatorial arrangements. I mean, all of these different strategies have in common the notion of gene regulation as a combinatorial landscape with some few elements, transcription factors, that combine in different ways and regulate accordingly many different genes, operons, and transcription units. Then the notion of transcription units and operons, I'm not going to go through details, but anyway, the initial definition was an operon conceived as one transcription unit, and the data has shown, as illustrated here, some elaborated cases where you have multiple transcription units overlapping, right? And not a single one, regulating all of them. I mean, transcribing all of them simultaneously. So that the simple solution was, in our definitions, to have an operon, simple and complex operons, simple operons with one transcription unit, and complex operons subject to multiple transcription units, working, of course, in different growing conditions. Finally, as we all know, gene regulation is a mechanism that is a way of sensing the environment and modifying gene expression accordingly, where environment can be either both the external environment that starts with a signal and gets incorporated, or in an internal environment through some metabolites that can be synthesized internally and also affect gene regulation. So the signal gets incorporated, and there's a signal transduction mechanism of one or multiple reactions that eventually generate an effector metabolite that will go and bind to the transcription factor, right? And alter and modify its binding capabilities to DNA. Some people talk about effectors as signals, and the use of the language is different. We propose to conceive this as an information flux, where the signal is always the one that is external to the memory, where let's say where the process initiates, and the effector is going to be the one that binds to the effector, in this case transcription factors, that will go and directly bind to DNA. So this gives you just a short overview of our discussions. What is the benefit of improving definitions? Well, I think that if we improve our definitions, we should be able to communicate better, of course, and to reason better. And of course, this will set the basis, if formalized into an ontology, for the interoperability of microbial regulatory resources. I finish here. Thank you very much. I acknowledge funding from UNAM, CONACYT, and NIH, and I am ready to take questions. Thank you.